Well, thank you very much, Emanuele and Telmo, for inviting me to this new format. I understand that you're starting, and I'm very honored to be the guinea pig to, to do the experiment. And I will be speaking in English because my Italian really is not good enough for giving a scientific lecture, but I understand there is simultaneous translation, and I think this is going to work very well. Now, as Telmo has already pointed out in the beginning very eloquently, there have been fantastic advances in the biosciences. He mentioned genomics and proteomics and all the omics, but there is also other advances in biomedicine. There is advances in imaging technologies, in measuring and quantification of biological data. And so, in fact, the empirical sciences in biology are producing an enormous amount of new data and an enormous avalanche even of data. And the really interesting question is whether or not this has any effect on our general understanding of life, or as we could call it, on our understanding of evolutionary theory. And I do not need, of course, to emphasize in this audience that theories are not static. Theories evolve, of course, and we may discuss how they evolve. Telmo has mentioned this. There are different philosophical views on how theories actually change and whether they change and whether there is rapid change or whether there is paradigm shifts or whether there's gradual. In fact, it's a gradualistic versus punctualistic debate as well. However, the interesting thing in the evolutionary sciences and in evolutionary theory is that the so-called modern synthesis is the standard theory still today, although it was created in the 1930s, 1940s, and you still find it as the standard paradigm in most of the textbooks, not all of the textbooks. There is a beginning of change, but mostly you see this. So Massimo and I and many others felt this cannot be the case. With all the empirical advances that have been made, that there should be no impact whatsoever on the theoretical underpinnings of our science. And so I am going to address this by first briefly recapitulating on where we stand. And of course, there is the Darwinistic core in evolutionary theory, and Darwin related these three factors to each other: variation of characters, the heredity of characters, and the multiplication of generations, and with the aim of explaining adaptation and speciation in evolution. And the primary factor, so to speak, that he coined was natural selection that acts on differential reproduction and so on and so forth. I don't need to go into the details in this audience. And then, over the years, this theory has been enlarged, and finally, in the 1930s and 1940s, what happened was the so-called modern synthesis, which took place through an inclusion of population genetics and systematics and paleontology and other things, as you know. But the primary goal of this theory was to explain variation at the population level. And the key symbol, so to speak, of this approach is the Gaussian curve, where you have characters distributed over a Gaussian distribution, and any factor that can impose a shift in the mean of the Gaussian distribution would be called an evolutionary factor. And so the core methodology of this, of course, is a statistical, mathematical approach to the distribution of characters, the Hardy-Weinberg equations, and so on. And essentially, evolution viewed at this population level regards the change in gene frequencies in populations as the core event in evolutionary biology, and it became the dominant mode of approaching evolutionary problems through population genetics since the 1930s and 1940s. But it contained no explanation for how the characters that are measured are actually produced or actually arrive. You know, the whole theory rests on a correlation between measurements of phenotypic characters and a statistical assumption about the underlying genetic change. Population genetics does not work with real genes. It works with statistical genes. And therefore, there is no mechanistic aspect in the standard modern synthesis theory that relates true molecular genetics to phenotypes at the morphological phenotypes. And therefore, we can extract a number of key features of the modern synthesis theory, and I do not even want to go and read through all of this, but what you can capture here is that this theory essentially is population-oriented. It works at the population level. It is centered on genetic change. It is a gradualistic theory in the sense that all changes that can be measured and can be explained would be small, gradual, variational changes that add up to the changes over time. It is an externalistic theory in the sense that natural selection from the outside, so to speak, of the organism is the only directional factor, the only factor in this theory that provides direction into which evolutionary change is going to happen. And I would add, it is an adaptationist theory in the sense that every character that you perceive in this species in the natural world would be explained as an adaptation to something. You know, there is no non-adaptational characters that the theory would address. So, theory doesn't address organisms, it doesn't address complex phenotypic traits, and it doesn't contain a mechanistic relationship between genetic variation and phenotypic change. It provides a statistical correlational relationship, but it does not, so to speak, explain how phenotypic change is developmentally and mechanistically realized through gene modification. So now this is where we stand in the 1930s, and surprisingly, this is where we stand in most of the textbooks even today. And so therefore, the interesting fact is that if you look on the internet, for example, and you look for evolution, you will still frequently find this definition of evolution is a change in gene frequencies in populations over time. And you also will see that this view is very explicitly defended by representatives of, let's say, the population-oriented camp, like Jerry Coyne here, who in this paragraph written in 2010 essentially says we have natural selection, we have the mathematical tools to analyze this. We will have, in addition, we will have genomics, but essentially, he says here, this is enough. That's all we need, you know, and nothing more is required. And this is what Telmo was addressing, and this is why Massimo and I and many others felt this cannot be the case. If we have a number of new areas, new subjects, new concepts that have arisen, that a paradigm created 70 years ago, or more than 70 years ago, is still the standard theoretical underpinnings for what we are doing. Therefore, I'm going to address 4 issues in the remaining parts of my talk. And the question is, are there actually any new concepts, and which ones are influential in evolutionary theory? Then the question will be, yes, there may be new concepts, but do they influence the structure of evolutionary theory? How, you know, the argument is run. An additional question would be, has, through the addition of these new conceptual aspects, evolutionary theory expanded its explanatory capacity? Can it explain phenomena that were not explained by the previous modern synthesis? And I would like then, in the end, to briefly summarize what the principal differences really might be between the standard theory and the extended synthesis. Let's begin with the new concepts. Telmo has already mentioned some of them. I will very quickly go through a few examples, most of which are in fact are represented in our volume. This does not mean that these are the only concepts that have been developed in the past 20 years or so. But a few exemplary things I'm going to address. For example, genome evolution. You know, all of you are aware of the high throughput genomics methodologies that exist today, gene sequencing, we have full genomes and everything. And it became clear that significant portions of the genome can be duplicated or can be co-opted into new functions, which represents an entirely different picture of genetic evolution from the past, where single genes were thought to mutate and provide all the variation that exists out there. Genome evolution provides a totally different account of the possibilities of how genetics may evolve, and therefore have an influence on how we can think about, for example, about the gradualism inherent in the modern synthesis approach. By the way, one of the best books on genome evolution is by one of your colleagues, Giorgio Bernardi, which is really a very interesting book to read. And we can see that these kinds of molecular events at the genetic level allow for much more rapid instances of change than previously assumed under a gradualistic, continuous, little additional variation paradigm. Multi-level selection, you know, in the standard assumption, selection acted at the individual level. But in the meantime, it has become clear that selection can act on the developmental or on the gene infra-individual levels, and also on supra-individual levels, such as kin selection or group selection, you may all be aware of. there is a question of whether species selection is actually also effective, but kin and group selection in the meantime are very well accepted, and especially David Sloan Wilson, who was also at our conference, has advanced this concept very much. So also the selectional aspect inherent in the modern synthesis has been modified since the standard period. Inheritance. Classically, only genetic inheritance was accepted. But in the meantime, we have learned not only about epigenetic inheritance systems, which I'm sure you're all aware of, but also about behavioral and cultural inheritance systems, which all provide, different levels and different mechanisms of inheritance that affect the evolutionary outcome. And of course, epigenetic inheritance, and by this here I mean transgenerational epigenetic inheritance, has been demonstrated. And Eva Jablonka was one of the most prestigious defendants of this approach, has collected in her last paper far over 100 empirically demonstrated cases of transgenerational epigenetic inheritance. However, it is not clear whether this has played a very important role in evolution. This is an open question. I'm not saying that now we have to revise everything we know about inheritance. I'm just saying it is an empirical fact that epigenetic inheritance takes place. Whether or not it had a huge impact on how evolution took place is another question. There is, of course, a great deal of new concepts coming out of the science that Telmo has defined as Evo-Devo, evolutionary developmental biology, in which enormous advances have been made. For example, regarding gene regulatory evolution, as you all know, the regulatory genes, Hox genes, and so on, have been called. And there are some very, very interesting new aspects in this that also affect how we think about evolution. For example, the aspect that there is a great deal of conservation in our regulatory genome. For example, here in the evolution of eyes, it has been demonstrated very clearly by Walter Gehring and others that the PAX6 gene, which is the homologue of the insect eye gene, or eyeless gene, is conserved over a huge variety of totally anatomically different organisms, and is implicated in the definition of the general eye field, and then, so to speak, this homologous gene that is active in all these different is at the source of very anatomically totally different constructions of the eye. So the idea is not that all these eyes are homologous. They are anatomically quite diverse, but this conservatism of major aspects of the regulatory genomes across wide varieties of different taxa has been rather unexpected, because under the traditional assumption different anatomical structures would be regulated by rather different genes. So this is an important change. Another concept coming out of Evo-Devo is facilitated variation, proposed by Mark Kirschner and John Gerhardt, it counters the traditional assumption that phenotypic variation is purely random. It shows that development constrains very much what can happen in evolutionary change, but also, and this is their point by the facilitation, development through its strong interconnectedness of very different levels and aspects of the developmental processes can facilitate change because since developmentally these structures are interdependent, change in one structure automatically elicits the following of the other structures. So, for example, if you have change, let's say, in the skeletal system, all the muscular tendons, the vasculature, the innervation of the muscles follow automatically. Whereas under the traditional assumption, evolution would have to wait, so to speak, for fitting variation in the other systems, where variation in one system was independent of variation in other parts of the body. And then there was always the argument there is not enough time that all this fitting variation can come together. Well, facilitated variation shows this is not necessary, because of the interdependency and high interconnectedness of the developmental processes. Finally, also in Evo-Devo, you get new concepts about how very first body plans of multicellular organisms may have arisen. This, of course, is also a very important question because under the standard assumption, it is always existing traits that are measured to calculate the variation. But the question is, how do such traits arise in the first place? And one of the models for the origin of multicellularity also comes from Evo-Devo, for example, proposed by Stuart Newman and Batt and others, where they show that first unicellular organisms have certain properties of adhesiveness, let's say, and other interacting molecules that under given conditions come together, and they only can form a certain limited array, a certain limited number of structures, such as balls of cells or hollow balls of cells or elongated rods of cells, or then depending on interactions with other molecules, certain periodic structures or whatever. And so these letters you see here in between, they designate so-called dynamical patterning modules. These are molecules that provide cells with certain properties under which they can interact. But the point is that, for example, if we take the adhesive molecules, it doesn't matter which molecules these are, the outcome of the possible forms will be the same. So there is only a few combinatorial ways under which you can associate cells. There is not an endless number of variation. And therefore, this concept speaks of generic forms, forms that will automatically result every time cells come together, independent of what the underlying molecular properties of these cells would be. Then, of course, once you have a certain standard repertoire of generic forms, natural selection can set in and can start playing on these forms and producing variation and selecting certain components of this array. And finally, one of the last things I'd like to address with regard to the new concepts is innovation. The Evo-Devo recognizes that next to variational change, there is also major changes in body plans that you can call innovations. And they arise under particular environmental conditions. David Jablonski, for example, has shown that the rate of innovations depends very much on the environment. And for example, in these post-Paleozoic orders, he shows that onshore organisms have much higher rates of innovations than offshore organisms, for example. And we have made in developmental biology, and again this comes out of Evo-Devo field, a number of assumptions about how such innovations can arise developmentally. And one of the major issues here is the threshold properties of developmental processes. That means if you have natural selection acting on developmental processes, it will bring such processes to a limit where there is a switch into a new homeostatic relationship, or the organism cannot cope with it and will die. But this threshold property of development, we think, is one of the major reasons how innovations can arise, such as innovations in the skeletal system, for example, where you have the whole gamut of molecular mechanisms already in place in order to form cell condensations and cartilage and other aspects. But when you have selection acting on the proportions, for example, of a limb, you get new cartilages for free, so to speak, because the whole reaction system is there, and if a threshold size of the morphogenetic field is reached, it will add a new cartilage by itself. You do not need to select for it. So finally, of course, phenotypic plasticity is an important concept also that has been developed. That has been known already quite a long time ago, and it is the idea of how environmental factors actually are translated by developmental reaction norms into distributions of phenotypes. This means, or this explains how the same genotype can result in rather different phenotypes depending on how the reaction norm in development works. And this has already been assumed under the standard assumptions, but what has been added to this primarily by Mary Jane West Eberhard is the idea of environmental induction, which means that changes in populations of species do not only arise through natural selection, but they can also arise through an environmental impact directly on the development of the species and on the development, on the ontogenetic development of the representatives of these species. And selection would set in only secondarily to fixate and stabilize the outcomes. But because these environmental factors would have the same effect if they are maintained on the representatives of these species over time, fixation doesn't need to set in immediately. There is time for natural selection to act, but so Mary Jane West Eberhard argues that in addition to natural selection, direct environmental induction can also be the initial agent, so to speak, for evolutionary change, which would be a quite revolutionary statement if it is further supported. Okay, so what do we learn from this? We could say that the modern synthesis has proposed a number of concepts. In the center here, you would see represented the Darwinian factors. In the blue ellipse, you would see, so to speak, represented the factors added under the modern synthesis assumptions, such as Mendelian inheritance and population genetics and so forth. And we could simply say there have been a number of new concepts that have been added, and we now recognize more factors in evolutionary theory. So what? This is fine. It's a simple expansion of certain details. And the question now, however, is, does this affect the structure of evolutionary theory? Is there any difference on how we think now of the interaction, about the interaction of the different processes that take place in evolution? And therefore, I will go to the second topic on altered theory structure, which I think comes more to the core of what we're interested in here. And I will try to present this through a schematic depiction of the interactions that take place in evolution. However, of course, you will understand that any schematic is a strong simplification. In fact, in this case, it's a model of a model, and therefore very simplified. But I think it will be possible to demonstrate what I mean. If we start out from a standard diagram of how the modern synthesis argues, you will see represented here the environment, and here are the populations of species, and we have gene pools, and we have populations of phenotypes, and between those we have genetic inheritance to the next population of phenotypes, and the next gene pool B, there is time is acting on this scale, and here is the environment, and natural selection would be acting from the outside, from the environment, which could be other species, of course, on those populations of organisms. That is, if you like, the standard reasoning in the structure of evolutionary theory. Now, under the assumptions of several of the concepts I have mentioned and others I have not mentioned, we could say it has changed in a number of ways. One is that we do no longer speak simply of gene pools, but rather of developmental systems pools, which is quite different, because developmental systems have all the properties I have mentioned, reaction norms and so on and so forth, threshold capacities, constraints, all these things. So this would change already the primary assumption about gene pools. The second thing is we do not longer speak simply of populations of phenotypes, but of population of plastic phenotypes, which have multiple possibilities of reacting to environmental change. Thirdly, there is no longer simply genetic inheritance, but also epigenetic inheritance happening between these generations. Fourthly, there is not only simple, single, individual selection, but we speak of multi-level selection, as I've explained. Fifth, there is not only an action, perceived from the environment towards the populations of organisms, but populations are actively reorganizing and influencing their own environment. Think of anthills or termite mounds or cultural effects. And this is a process I've not addressed, but this has been termed niche construction. And so populations actively change the environment, which itself becomes the selective environment for the next population, for the next generation. So therefore, there is not only inheritance going on at this level, but there is also inheritance going on at this level, which we would call niche inheritance. So organisms reorganize the environment, which becomes the selective agent for the subsequent generations, and is a requirement for the next generations to actually live in this. So what I mean to say, and then there is finally one more aspect, which is the one I've explained with Mary Jane West Eberhard's view, which is the possibility that environmental factors directly act on the developmental systems, modifying development through, let's say, physical aspects, temperature or gravity or humidity or whatever, and then selection can act on the reactions that are provided by the developmental systems. So what I mean to say by all this is that our understanding of the structure of evolutionary factors has become much more complex over time. If you compare this image now to the one I've shown in the beginning, you know, it is clear that that this was in this standard view there were much less factors and much less interactions involved rather than in this view. So there is a more pluralistic account now with many more causal interactions taken into account, and of course I do not draw all of them in here because it would become rather complicated. But this is maybe the effect that we can show on the structure of evolutionary theory. Explanatory expansion. Can we explain effects that have not been explained under the standard assumption? I'd like to start out and again bring only a few examples, such as discontinuous forms of change, structural complexity, morphological novelty, homology, conserved body plans. These were phenomena in phenotypic evolution that were not even addressed in the standard account. Of course, one cannot say that now all of this is fully explained, but at least it becomes within the reach of explanation and can be addressed. Let me show you some examples. For example, one of the great enigmas under the standard assumption is the genotype-phenotype relation. How do genes actually translate into phenotypes? And there is a paradox here, which you can simply describe by the relationship between gene numbers and the complexity of the organisms that result from them. For example, as you probably all know, the developmental model organism Caenorhabditis is a worm that consists at birth of 558 cells. Exactly 558 cells. You know, it will not change. So this is a very deterministic developmental system in which every single cell has, over the entire developmental process, has its particular place and role in the development of it. And compare this to an organism like this mammal here, which consists of billions of cells and hundreds of different differentiated tissues, but the gene number that regulates these two different organisms is roughly the same. So now, where does the complexity come from? And of course, it comes from the regulatory interactions between these cells. So it's not like under the standard assumption where you get, you know, new genes for new characters and you need to wait for the variation of these genes in order to, over a long period of time, produce these new forms. But rather, we know that the evolution of complex phenotypes is regulated by very similar genomes. And you know all about, you know, orthology and homology and collinearity and all these aspects that are part of the molecular genome. I also would like to address one of the aspects that we have been working on ourselves, which is the new explanatory possibilities for what we call novelty. innovations in the phenotypes that are not directly related to innovation at the molecular level. And there is a number of different types of innovations that one can discern, such as the primary multicellular assemblies I was showing with Stuart Newman's work. But then there is two ways in which novelties can be introduced into existing body plans. And here we must distinguish between what we call discrete new elements that had no forerunners. If you, let's say, evolve a limb and new elements are added to the limb, versus dramatic exaggerations or individualizations of existing elements that have also been called novelties, such as the narwhal tusk, which is, of course, an incisor tooth which is extended extremely long. And these kinds of variational novelties, as we call them, they are explained under the standard model because it's a variation that is continued and continued and continued under the assumption of natural selection. You get, the addition of new elements to an existing body plans is something that has not been addressed in the standard theory, because the standard theory is always occupied with measuring what exists already. It focuses on the variation of existing elements, but not on the production of new elements. And here we have made a number of cases on how innovations arise, and I have already mentioned that a certain type of innovation might be related to so-called threshold effects in development. That is a long story to tell, which I can only shorten to 1 or 2 slides, but that such threshold effects exist, you can simply see on these pictures already. You know, if you look at these limbs, which come from Australian skinks that have undergone different levels of limb reduction, you know, because they assume a snake-like behavior, and in many different species of those skinks, the limbs become reduced and reduced and reduced more and more. But as an effect of this reduction, the limbs start losing digits. And you see the sequence of digit loss in these limbs. There's a particular sequence, I don't want to go into this, but what you can see very clearly is that it is not a variational reduction in which the distant phalanges become reduced first, what you would assume, and then more and more phalanges will be reduced slowly. No, it is complete single digits that are lost at once, in one step. You know, here it is digit number 1. Here, in addition, is digit number 5 is lost. Here, the second digit is also lost, and if 1 digit remains, it's always digit number 4. So these are threshold effects that you also can demonstrate experimentally. If you reduce the cell number in the developing limbs, you get also complete losses of single digits, not the gradual variation that you would assume will take place when you reduce continuously a certain parameter. You would expect that the phenotype follows also in a continuous variation. It's not the case, and the limbs are a very good example, and here you see the thresholds at which certain digits will disappear. The same, by the way, takes place in the addition of digits. There's a number of mutants in mice and other animals, even in humans, where you get added digits. And it's not just a little added bony element, a little phalanx or whatever, but complete digits are added. And even in fossils such as Acanthostega, which is known to have 8 digits, you know, you can see that complete digits are added. So both the loss and the addition of this phenotypic character happens in jumps. It's not a gradualistic kind of event through continuous little variation. And these are effects of development, as you can show through developmental experiments. Stuart Newman and we have, for example, constructed a model of limb development that can show and demonstrate these effects, which is based on a number of molecular factors interacting in limb development between the chondrogenic cells that take place. But you can put this in a simulation and you can show how digits appear and disappear simply based on those cellular interactions. By this I mean that the developmental approach to evolution makes a distinction between continuous, gradual, small step variation, which is the standard model where you have some individualized character that exists already, you know, and then there is variation, and natural selection acts on this variation, and the outcome of this would be an adaptation of a particular character. In development, we see that selection can act, or natural selection or environmental induction even can act on some developmental property. There can be a threshold response of this system, and the outcome would be either a deletion of a character and the complete disappearance of something, maybe the death of the animal, or if the reaction can be sustained, it could be a novelty that appears in the body plan. And then, of course, you can start from here. And you can start, you know, the novelty can be assimilated, fixated, stabilized, routinized in development through the molecular mechanisms at hand, and then you get the standard picture on how it can be further refined and varied over time in populations. So that is one of the outcomes of Evo-Devo research. Finally, what would be the real principal conceptual differences between the standard model and the way in which we are thinking about evolution today? By the way, nobody is calling for a new theory. Neither Massimo nor I, nor the other proponents of this are saying we need a new theory of evolution. We're just saying that all the models, concepts, and factors people are working with today are already different from the time under which the modern synthesis has been constructed. And we only provide a kind of umbrella name for this new level of theoretical approach, but it doesn't mean that anybody says here we need a new theory. It just is not possible to do all this empirical and theoretical research with new concepts, but call the standard model under which this is done still by the name that was accurate 70 years ago. So the principal difference is I provide in a dramatically simplified table, ridiculously simplified table, because every one of these terms here could fill a book, you know. But since it cannot be done for several hours, I'd like to point out that some of these major differences. One is that the approach, the theoretical approach under the assumption of the modern synthesis was primarily a statistical correlational one, whereas in the extended synthesis it is a mechanistic approach about how genes provide properties to cells that reorganize and can translate into patterns, and patterns that form structure, and structures that form organisms. This is a very different approach from the correlational approach. The focus in the standard theory has been genetic primarily, whereas I would say in the extended synthesis it is an organismal approach that takes into account the environment, it takes the properties, it takes behavior, it takes development, all these aspects. The evolutionary phenotypic change was under the modern synthesis assumption was entirely viewed as a variational change, whereas in the extended synthesis approach, we accept not only variation, but also these emergent properties that arise through development, some of which I've tried to explain. The rate of change under the modern synthesis assumption was by necessity, by requirement, was required to be a constant rate of mutational change and a constant rate of variation, whereas the modern synthesis, the extended synthesis also allows for discontinuous forms of change, which I've briefly explained. Natural selection as an explanatory agent had a principal key role in the standard theory. In fact, as I've said, it was the sole directional factor that was active under the modern synthesis assumption, whereas in the extended synthesis, natural selection becomes a boundary condition, which is always there, which is continuously acting. However, it is not the factor that provides the specificity of evolutionary change. The specificity of the resulting outcome of natural selection would, under the new assumption, would be provided by the developmental system and its reaction norms. It can produce only certain responses. Natural selection would be the boundary condition under which these developmental reactions are released and then can be further refined. The causality under the standard assumption for evolutionary change, which is more or less what I've said already, was entirely external, the environment, natural selection, other organisms, other populations, whereas under the extended synthesis assumption, the causality is, in addition to the external forms of natural selection that take place, are by the internal rules and capacities of the developmental systems. Therefore, expanding on this last point here, my final comment in a more philosophical point of view, which I believe is because the groups, is Telmus groups, I would like to briefly mention this. There has been, if you view this in a much more general philosophical way, there is a change of explanatory weight in the new forms of the evolutionary theory that I'm proposing here, in the sense that the buzzword, so to speak, under the standard modern synthesis assumption was contingency. Contingency as lawfulness depending on large amounts of chance. Chance events played a very important role, and the explanation revolved around this contingent results. Maybe many of you know the metaphor of Stephen Gould's metaphor of when the tape is replayed twice, the outcome will not be the same. You know, this was illustrating contingency. And I would propose that under the extended synthesis, we also include what Stuart and I have been calling inherency. And inherency is what will always happen. It will always happen on its own accord because of the internal constraints, evolvability, the emergent properties of developmental systems that provide certain rules under which a change can take place. And therefore, according to us, if the tape would be replayed twice, we would say the outcome is not entirely different. It could be different, of course, because of environmental effects such as meteors that cut out complete populations and so on. But what would be generated evolutionarily, according to us, would be recognizably similar. And that is where I would like to end my presentation, I would like to add that, of course, all this was a very brief recapitulation of what we have written in the book, or what the authors, the contributors, have in fact written, and more can be seen. This, by the way, there is a companion to this volume, which is a re-edition of Julian Huxley's standard work, Evolution, which in fact coined the term the modern synthesis. And Massimo and I, we have a foreword in this, but otherwise we are not responsible for this volume. Thank you very much.